Nathan Pearson is from QIAGEN, and QIAGEN is a sponsor at the GET conference, and we're very happy to have him here. So actually, I'll just take the computer away.
Scoot that one right in there. Thanks. Thank you, Jason and George, for having us here. We're really proud to be here working with you all to help make your data more useful scientifically. And I want to talk today a little bit about our effort to do that with a new outreach called the Empowered Genome Community. We talked about this a bit yesterday at Get Lab, so this may be familiar ground to some of you now. But for those of you who weren't there yesterday, I want to introduce the effort and so you know a bit more about it. As George noted, we've really grown in terms of the participation here at these conferences for Get over the past few years. And since early ones where all of the sequencees in the world could fit on the stage, things have changed a lot. So I want to talk a little bit about another precipitous graph in addition to the price one that George showed. This one is a poignant cartoon from XKCD on the number of people who've walked on the moon. So these are the lunar astronauts projected to die off over time by actuarial tables if we don't go back to the moon. So this is a reminder of where we've been scientifically and where we could be again. If we contrast this sort of sad, poignant graph with the people who've been sequenced, that's changed a lot. When I started interpreting genomes for people about 4 years ago or 5, there were fewer people who had been sequenced than had walked on the moon. And that's really changed incredibly over the past few years. The graph has now skyrocketed off the launch pad here. And if we look at it in Venn terms, the waning gibbous of moonwalkers compared to the number of people who've been sequenced, and they don't overlap yet. We should sequence a lunar astronaut. But more of them have been sequenced through PGP than through any other program, of course. And there are other programs growing, which is fantastic to get this kind of data back. But the amazing consents and phenotype data in PGP make this data particularly valuable. Those of you in PGP, though, may know that it's tough to get a lot back from your data yet, for many of us. In terms of long-term prognostic insight about what our genomes will mean for healthcare over the course of life from birth to old age. To get to that point is going to be tough, of course. Typically today we get back a lot of raw data that may be hard to wade through. Perhaps a report that's sort of weakly prognostic. I think of these as a stack of kind of frozen waffles, where we don't know much yet, and it's often a sort of frozen vision of our genome at a given time and place in science. And perhaps we get an application for browsing through some of that data in a limited way. And what we learn is often a fair bit about ancestry that we could also learn typically from common variants on a SNP chip. We learn a bit more about what I call drugs and mirrors, and I don't mean cocaine here necessarily, but other kinds of drugs we may respond to in the course of healthcare. That's an important front of insight for many of us. And we learn a bit about the genetic basis of what we see in the mirror, why we have the color eyes we do, et cetera. We can often trace some of those things to particular spots in our genomes and understand them better. We may learn a little bit about carrier status and or personal health. But we often learn a meta lesson, which is what's missing. The big expectation we may have had going into getting our whole genome sequenced, all that data must tell us something. Well, it's a very, very hazy crystal ball, as many of us learn. And in order to make that one genome more reliably informative, we're going to have to ask a different set of questions over a different time frame. The reasons are that, of course, health is complex, lots of causal factors explaining why we get sick when we do. Rare variants that play roles in that health, they abound in our genomes. It's important to remember that each of us has more novel variants in our genome, never before seen by science, than there are ClinVar variants total. So remember that. You've got a lot of really interesting data yet to be tapped that can help shorten lists for sick people soon. And importantly, that our data are still scarce. They're still very valuable. There are too few genomes yet with good phenotype information that have been sequenced. So we saw those 3 challenges. We wanted to make a program to help meet them, sequences to let them mine more data from more insight from their genomes, and that entailed comparing many genomes and getting those to researchers who could actually do the number crunching to actually find trace disease to genomic variation. So I want to talk a bit about the effort we've devised to do that, called the Empowered Genome Community. There's another Owly quick URL you can go to right now to see it. The idea was to give sequencees access to a really good online platform for interpreting their data functionally and for comparing genomes to each other. To help us ask the key question today, which is not quite yet, what can my genome do for me? We'd love it to be that, but it's really not. It is more, what can our genomes do for humanity together? And in order to answer that question better, we let sequencees access the platform where they can leverage the knowledge base that Ingenuity has built over the past 15 years of what do we know about what particular genes and other parts of our genomes do functionally from people and mice and other organisms. And importantly, compare those data in small numbers, so a few genomes at a time or hundreds of genomes at a time, to each other in a comprehensive way that statistically finds patterns that tell us where disease or other phenotypes may trace to. And importantly, all the sequences here, all of you genonauts, retain the privacy of your own private data. Now, PGP genome data, of course, is public. There are other things you can annotate that data with in your accounts that will remain private to you unless you choose to share them. It's a very shareable platform if you wish, in the spirit of PGP, but it's also respecting your privacy for private data. We have a proof of principle study, which is the open myopia collaborative analysis of myopia that I encourage you all to go see. We've taken 111 PGP genomes that have been annotated with respect to eye phenotypes, thanks to Madeline's surveys that are so important to making the data useful. And we've done a case control analysis for myopes versus non-myopes, and shortlisted about 25 genes that look enriched for rare, functionally interesting variants in the people with myopia and not in those without. Importantly, this is not a published study. This is an ongoing crowd refinable study that you all can go onto in your accounts and variant analysis and help us refine and publish together. Anybody who makes substantive insights to this study, who grapples with the data with us in good faith, will be a co-author on the paper that eventually reports what we do or don't find about the genetic basis of myopia, particularly rare variants, in this still fairly small study of 111 people. We encourage you also to work to give more data to Madeline and the project overall so that we can make the study, refine this study and others in future. Here's a snapshot of what variant analysis looks like. You'll all get to see it in your own accounts if you go to those URLs or talk to me about it. And we're really proud to be supporting PGP and working with PGP in this. Summing up, joining, if you can bring your genome, you're all welcome in the Empowered Genome community. You have your own account there ready for you. As soon as you register, your genome will be there waiting for you and you can start to explore it. If you don't have your genome yet, you can still bring your mind. Your mind can help us understand that myopia data and other kinds of studies we do in future. And you can also, if you're a researcher, bring us your wish list. So what kinds of phenotypes interest you? We can put people in touch with others in the community who may have or not have those phenotypes and may have data useful for you. Thank you so much for listening and for being here with us.